So hello everyone again, Microbehunter here, hi, and welcome again to another Saturday live stream. I'm Oliver, and I've seen that many of you, some of you have already joined over the chat. Today I would like to put some of my water samples again under the microscope. The last time I did that was around half a year ago. And you can already see a video here on the side. This is a time-lapse video of one of my samples, which is over 1.5 years old. And I would like to talk about that. Somebody requested that I wait for a few seconds before starting to talk because there has been some delay in the sound. So I do hope that you're able to hear me properly. And and of course there are already people who are wishing Happy New Year. Also from my side, of course, a Happy New Year all around the world. Okay, you know what I'm gonna do? I'm gonna start off a little bit differently today. A little bit something unexpected happened before I actually start jumping into the actual topic. I just hurt myself in the finger. Yes, actually, I did. I'm bleeding a little bit. So look what I got here. I got a small drop of blood, and I have to take advantage of this. So this is basically I just accidentally cut myself and says, well, why not just put the drop of blood under the microscope first? Yeah, before I actually get started with the actual topic, because. Yeah, why not? Yeah, and usually what I recommend is to look for the white blood cells. You make a very, very thin film of blood, then you are able to see also the white blood cells. And I'm going to just do this for right now because it will take a couple of minutes for more people to join in. Yeah, and here we can already see at low magnification the white blood cells, but I see that the frame rate is not very good. So what I'm going to do is I'm going to adjust the camera a little bit, the camera settings. Going to make it dark, so this, and I'm going to turn up the light, and here we go. Here we can you can already see a whole bunch of air bubbles, and those tiny little dots that you see, these are the red blood cells. Just a little bit as a little warm up for to wait a little bit until more and more people join in here. Those large round circles, these are a little bit air bubbles. So this is how it looks like. And now the bleeding also stopped. Let's turn up the light again. So that's it. Kind of shows very nicely the shape of. I got the wrong setting. Just a second. Here we go. Okay, and let's open it up a little bit. So these are red blood cells. Okay. I like to have a look at them. And when you wait a little bit, you're actually going to see that some of them start to stack up a little bit. And that's called rouleau formation. This is as the blood sample starts to dry up. Okay, but I think I'm just going to get now started with the actual topic. The rules of the game, you're probably already familiar with. That if you essentially have any questions, then please do post them in the chat. Maybe you would like to include an @Microbehunter or @Oliver in front, so that essentially I know that the question is addressed to me. And I would also later on like to have a look at some of the permanent slides that I made last week. If you've joined in, then you probably remember that I've tried out different mounting media to make the permanent slides. And we're going to have a look at this today, how they developed. And if you missed last week's a live stream, you can find it, of course, in this channel. So all of the live streams are recorded. So these are some of the slides that I made last week, and we're going to have a look at them because there is going to be a little bit of a difference visible depending on the mounting medium. Okay, so what I'm going to, another thing I wanted to show you a little bit before I get started. Look what I made. This is, we've got now a laser cutter. So I made myself a little box here to hold the slides. So I think that's also, besides a 3D printer, laser cutters might also work to make microscope accessories. But that's going to be basically a different topic. So what I'm going to do now is I'm going to have a look at this specimen over here first. And I collected this in summer last year, so over 1.5 years ago. And this is a marine water sample from northern Germany. And what you see here is a time-lapse video. So there are still a lot of microorganisms alive and happily moving. And why time-lapse? Because the movement sometimes is too slow to be seen. I just want to show you now the sample. Here it is. It's basically a marine water sample, and I've got some sand on the bottom here. Essentially what I've done is I've added a little bit of some of the sand water mixture on a microscope slide and looked at it. I'm quite happy that it was able to stay quite consistent over the last couple of months. It's marine water, so it is a little bit more stable than fresh water. And I'm going to simply show you now how this looks like under the microscope. And over there I've got 1, 2, 3, 4, 5, 6 other samples that I would like to check because I'm now in the process of cleaning up everything a little bit, and I want to throw out a few of the samples if they're not needed anymore. But before I do that, I would like to have a look at them. Now I like to use these pipettes here. However, the tip is a little bit too small. So for this reason, I recommend that if you want to take up a sample that is a little bit larger, then I recommend that you cut off the tip. Okay, here, this basically makes sure that the tip is a little bit wider, and this way also larger samples can be taken up. And I can just go down into the sample and just take up a small amount. Goes over here on the microscope slide. I have to check, not a lot of sand. If it's too small, then of course, you might not be able. I see there's a lot of sand here on the tip here, but I was not able to pick up a lot of sand here because, of course, the sand is heavy and likes to fall down. But now I got some. Okay. So let's have a look at this. It's important to kind of spread it a little bit because the sand otherwise is going to be covered up, and of course a cover glass goes on top. I just recently cleaned those, so it shouldn't be a problem. And now everything goes under the microscope, and then we're going to have a look at this here again, the red blood cells, which I'm going to remove. And now let's have a look at the sand sample. And again, these are, it's way too bright. Oh, look, interesting things here. This already looks quite nice. However, when we zoom in, then we're able to see, of course, not only the sand grains, but between the sand grains, we might be able to see an occasional movement as well. Usually these are diatoms. So I'm going to activate the arrow again. To kind of maybe point at some of the objects. And we have to look carefully. There was something moving over there briefly. But usually the movement is so slow that you're going to have a difficult time seeing it. And for this reason, what I've done is I made a time-lapse video, simply recorded for a couple of minutes, and then I was speeding it up, and this is when I was able to see the movement. Okay, so but let's go up a little bit more. You can see over here, sometimes there are some diatom shells as well, like those over here. Look, there is a little bit of movement over here going on, so you might want to have a closer look at this. So I'm going to zoom in there a little bit more. So let's center it. Okay, let's go up. Let's turn up the light again. And here we go. And you see that sometimes the whole sand grain is now moving, because sometimes the diatoms that are connected to the sand grains, they're kind of pulling along the whole sand grain. This one over here as well. So I'm showing this to you simply to make it clear to you that simply because you do not see any movement right at the first second or so, it does not mean that there is no life there. Sometimes the movement is simply too slow, and therefore it's difficult to see. So I'm quite happy that over 1.5 years the sample survived. Basically what happened is I let the water sample stand on my window, so there is light is able to reach it, because of Of course, those diatoms, they do photosynthesis. But then, of course, there was the problem that some of the water evaporated, so I had to always keep on adding a little bit of fresh water again. Because if water evaporates, then, of course, this increases also the salinity, the salt concentration, and you don't want that to happen as well. Yeah. So that's kind of the first thing I wanted to show you here. That essentially there is, quite a bit going on here. Again, let's compare this again with the video that I just showed you. Yeah, this is 10 times faster. Yeah, and I move forward a little bit, and this here is now 5 times faster. Yeah? It's the same sample which I just recorded just a short time ago. Yeah? So this sample I'm definitely not going to throw out, but I'm going to keep it, we're going to. I'm going to keep on checking periodically how it's doing. Yeah, so. Quite nice to see, and I'm quite relieved that the specimen was able to survive that long. Okay, so what I'm going to do now is I'm going to, as always, interrupt myself every now and then to answer some of the questions. So the very first comment is this Christmas I received a microscope and I have already viewed tardigrades and rotifers through the moss samples, and was wondering if you have any ideas of what else to look at. Well, there are lots of things that you can look at. If you basically don't have permanent slides, then I would suggest that you get a box of permanent slides as well. So then you can always look at something without having to prepare anything. But otherwise, I would simply recommend that you make yourself some tiny little projects. So if you have already observed rotifers and tardigrades, start taking pictures of those. Make yourself a collection of videos. Yeah? Share them online. Keep yourself a little bit active. Yeah. I pointed out during the last stream that I had a video but no sound, so I do hope that the sound works now. Maybe there was some kind of a delay, but I think nobody complained up to this point about not being able to hear me, so I hope that it works. And again, if for whatever reason the stream becomes interrupted, then I'm going to restart it. Yeah? A couple of months ago this actually happened. All of a sudden, for whatever reason, it got interrupted. Yeah. So, and that's what I do sometimes when I get cut. Yeah, I was just basically, accidentally cut myself a little bit and directly put it under the microscope. Yeah, I'm very curious about this stream. A local pond is hibernating, not much going on. Well, there is plenty of things going on. What I recommend is that even in wintertime, collect a few sediment samples from the bottom of the pond and let it stand at room temperature for a couple of days. This will allow some of the microorganisms to regenerate, and they will start to reproduce a little bit, and then actually you have something to look at. Yeah, but it is generally correct that during the wintertime, of course, everything is much less, but there is still enough to look at. Yeah. Okay. Yes, I was asking because I know that some of my students are also actually joining in. For those of you who don't know, I'm a biology teacher, and that's why I was asking. Who was joining in? So, And so hello from all around the world. Again, do you mind to put some microbes in your blood? Blood sample. Recently, I've learned some interesting capability of red blood cells that they might be able to break down microbes. Actually, it's not the red blood cells, but the white blood cells. As a matter of fact, I am also planning to do that. It is indeed, but it does take some preparation time, and it's a time lapse. So what you can do is you can take some microorganisms and mix them with some blood, and then you can actually sometimes see that the white blood cells attacking the microorganisms and breaking them down. And this is possible to observe under time lapse, because the white blood cells, they do move kind of slowly, and this will take a long time, half an hour. And therefore, I'm going to work on that, and then maybe I'm able to present either on the channel or in the live stream. I'm going to present some of the some time lapse where maybe white blood cells are able to break down microorganisms. Yeah. Why do the blood cells stack up? Why don't they just dry out? Well, the stacking up is called rouleau formation. It is a natural process because the red blood cells basically have a surface. They stack up as the blood dries, and then the flat surface has a larger surface than the sides, and therefore they will start to stack up. And you can see that actually happening in different parts of the slide at different times. So usually those places where they start to dry up first, this is where they start to stack up. But when the blood film is very thin, then there's not enough space for them to stack up. So you see there are a whole bunch of variables involved here. Yeah. Did you keep them closed, lid? Yes, I did actually keep the lid closed. And I think I'm just going to, there are lots of questions, which is nice, but I do need to explain this a little bit. It is, you cannot see this maybe, I'm just going to flip it over, yeah, it's the camera. You can see that there is a little bit of sand in here, this is all marine water, and there is lots of air here, okay? And that is perfectly fine, and when you keep it closed, then there is still going to be enough air in here, and it's not going to become anaerobic. There is a problem, and I've had this already before as well, is if you use those collection tubes, okay, and if you fill them up, or these are centrifugation tubes, and if you fill it up with a water sample all the way to the top, and you close the lid, then it's going to turn bad very quickly. Okay, it's going to start smelling like sulfur, it's going to go anaerobic, it's going to die off. Right? Anaerobic bacteria are going to take over. So, but if you leave enough air, then actually you can transport it quite well. Yeah, so so don't make this mistake if you go out collecting samples. Always make sure that there is, you can close it off for a couple of days, but make sure that there is enough air in there. Don't fill it up completely. I already made that mistake once. Yeah. So what I'm going to do is I'm going to just move on now. I'm going to put the next sample under the microscope. Okay, just a second. I have to mark this. So I move this away. So this was, oh, no. I just wanted to show you something else. I prepared another video. This is basically the place where I collected this sand sample. Okay, this is northern Germany, Wattenmeer, the Waddensee it's called. This is, basically they have a very, very nice area. Their tides are very strong, and you could walk out for kilometers, and then this is when the tide is basically coming back. So within 20 minutes everything's going to fill up, so you got to be a little bit careful when you walk out there. But this is the place where I collected the sand sample. Okay, so this just wanted to show you a little bit the context here. Okay, so here we are again with the sand grains, and sand grains and the microbes in the so-called the interstitial spaces. So the spaces between the sand grains are called the interstitial spaces. Okay. So what I would like to do next is I would like to have a look at this one over here, at this sample which I collected a couple of months ago. There are some algae in here, there's some sediment in here, and in this sample I was able to find a lot of worms, Iolosoma, and I don't even know if they've survived so long, and also Paramecium buerzeria, which is a single-celled protist which contains green algae as endosymbionts. Now this was a couple of months ago, so I don't know to what extent it's still there. So I'm going to take some of the algae that I've got here and some of the sediment down here, and I'm going to put them on the slide. And I'm going to put this one here I have to trash, and I'm going to take myself a fresh tip. Okay, fresh tip, and I'm going to collect some of the sample from the bottom here of the tube. And let's have a look here. Small one over here, and the algae I have to pull out with some tweezers, and I don't know if I'm not able to get in there. So what I have to do is I have to go in with I've got a little wooden stick, and hope I'm able to reach some of those algae. Yes, here they are. And what I usually do is I put it in here. I take some of the water sample, add it on top here as well, and let's have a look at this, at both of them. Okay, and again, I am not quite sure to what extent this will be successful or not, but there is a worry that I have, and that worry is that I do not have enough water on here. So if this is the case, then I might end up crushing the specimens. So I'm going to add a little more crushing, because if there's not enough water here, then the cover glass is going to be too close to the slide, and then this might actually crush it. So here, That's from the bottom of the tube, and this one is the algae. You can see that it's pretty thick, the specimen. So let's have a look. Honestly, I did not look at this for several months now, so it is going to be something that I also do not know how well I'm able to find something. So oh yeah, it's way too bright. No, it's way too dark. Okay, where are the? Ah yeah. You know what? Let's start with the low magnification first. Let's do this here. Ah yeah. So okay. This was definitely way more active a couple of weeks ago. So here I do not see, well, I mean, obviously the algae themselves are interesting enough. Okay, but let's have now look at the sediment. And ooh, I do see something. Yeah, that's my favorite worm. Yeah, so evidently, yeah, the sediment is quite alive, or at least quite. I mean, this worm is known as Aeolosoma. And very nice. Okay, I can talk a little bit about this guy here. Okay, so this is actually a nice example of an animal which is able to reproduce asexually, and it does this by dividing in half, and we can actually see this process already happening. Okay, so if you look carefully, and now this is going to be difficult because the guy is moving around so quickly. Let's have the arrow. I want you to have a look at this part here. Do you see this here? It looks a little bit more narrow. There is a new head forming here. So after a couple of days, this back part is going to split off and it's going to become a separate worm. I've actually observed this a few times already. Yeah, this is basically, you see that the worm is starting to fragment and a new head is forming here. That's why it looks a little bit irregular. And after a couple of days, you're going to see that the head will become larger and larger, and then it's going to separate off. So this is actually a very nice way of seeing that. So this is actually a sign. Now you see it quite nicely. It's actually a sign that this sample I'm not going to throw out. Evidently those worms are pretty happy here. Yeah. But what I'm going to do now is I'm going to again go to a lower magnification. I'm going to check if there's anything else of interest here. So at least this worm here in the sediment. Maybe, I don't know if there's another one over here. But if I was able to already find it on the first try, then this is actually a sign that there must be quite a few of them in there. Maybe not on the slide here. Yeah. But definitely in the tube. Yeah. So, yeah, the green things here, cellular fragments could be algae, yeah. But this worm here is, I think, the one that we're all most interested in. Okay, so I'm kind of relieved that this guy is still around. Okay, so let's have a look at them, at some of the questions again. I'm going to quickly. You are correct. White blood cells will attack the microbes. However, I have recently learned that red blood cells might be able to break them down as well. Would like to see if the phenomena can be reproduced. That's an interesting thing. Yeah, I'd have to research this a little bit. Yeah. So what is the exact name of this animal? It's known as Aeolosoma. It is the oil droplet worm. Aeolosoma. Yes. Yes. If you actually, it's difficult to see now because the guy's moving around so quickly, but actually if you zoom in a lot and if you have a close look at the head, then you might actually see that, especially if it's addressed, some of them actually have a red oil droplets in the head, so red dots on the head and on the body. Yeah. It's an oligochaete worm. And what I've done as well in the past is oh, that looks like the shell of an ostracod, but it's not alive anymore. Look, it's even cracked open. Ostracods, these are basically water crustaceans. So this is broken open and it's empty. Yeah? What did I just want to say? Yeah, I was once feeding those Alliosoma with a little bit of potato starch, and you could actually see them eat the potato grains and the starch grains, and you could see how the starch grains are traveling through the digestive system. So I have to go down again with the magnification because I lost him. Yeah, so that's it's a little bit too bright. Yeah. So I don't know where it is. Yeah. So let me have a quick look. There's some algae are here again. Yeah. So it could be just because you do not see anything here does not mean that there is nothing more in the sample. So what I generally will do as well is maybe I'm going to take a small sample here and I'm going to try to make a so-called enrichment culture. An enrichment culture is I'm going to add a little bit of food and then usually microorganisms will start to reproduce. Yeah. So I completely lost it now. I don't know where it is. Yeah. I need to go down again here. Yeah, but you know what? I've got so many other samples that maybe I'm just going to move along. Yeah, but yeah, it's somewhere. Nope. Let me see. Now I want to find it again. Where did it go? Interesting. Yeah. No, it's not going to be far away. Yeah. Usually they're pretty large, and you're able to see them even without a microscope. Yeah, if you just make sure that it's on a reasonably dark background. So I'm going to move on. So this is basically the sample I'm going to keep. The next one is one which is 3 or 4 weeks old. This is a sample from the aquarium. Yeah, in the school we have we have an aquarium, and I collected some water plants, and I see that there is also a snail in there, or the shell of a snail. It developed. It was not there before, but evidently I also collected some snail eggs inadvertently, and therefore those snails also started to develop. Okay. So I'm just going to take a sample here, and let's have a look. As a general rule, if you want to do microscopy, always make sure that you take some solid material. Okay, this is way too much. And what I normally do in this case is always take a fresh tip. You do not want to cross-contaminate too much. Doesn't matter anyway. But usually what I do is I will take the tweezers and by dipping here I can see it, the specimen, you sometimes are able to rinse off cells that might be on it. Okay, this is way too big. So what I'm going to do is I'm going to take a small sample here, and the rest goes back. Okay, so a little bit of, I'm going to dry it, and let's have a look here. Normally, or not normally, very often, if you want to find interesting organisms, they will sometimes attach to a surface, forming a biofilm, and this is the reason why I took some of the algae here, and also some of the water plants. So let's see how this looks like. And again, here we go. So, here, that's a water plant, obviously. And we can see already some microorganisms floating around. Okay. And what is this here? It also looks like a worm. It's also an Iolosoma worm. Wow. Where'd this guy come from? Ah. So let's have a closer look here. Here it is. A totally different water sample, but same worm. Yeah. So So this sample also seems to be fairly healthy. Not surprising, because if you have any water plants, algae, anything green in there, then it will of course produce oxygen, because those organisms and those animals, they require a lot of oxygen, and plants will actually supply that. Yeah? So this is the reason why, yeah, many microorganisms feel comfortable, yeah, next to oxygen-producing plants. Yeah. So this is a little too heavily magnified, but I think you get the idea. Yep. Let's go down to low power. Okay, and I would like to see a little bit better what those are. Here. They're kind of hiding a little bit away. The difficulty is because the water plant is fairly thick, so that means they're able to swim in and out of focus vertically, and that means they're kind of also difficult to chase around a little bit. But you see that they like to stay somewhere always close next to the plant. Yeah. So let's see if sometimes you see them float away and then they turn around, because they will follow the oxygen concentration. Now this one went actually off here. Yeah. It looks like looks a little bit like a small rotifer. Yeah. Yeah. So let's do the following. Let's really go up with the magnification yet more. So maybe we're able to see the individual cells of the plant. Yeah, here we can already start to see the cells better. Okay. So yeah. So, okay, so far no questions for me. I have to make sure that they do not move off to the side too much here. But this specimen also seems to be doing fine. Okay, so this is actually a good one maybe to make an enrichment culture. So I'm going to take a small sample here, add a little bit more water, and then add a crushed wheat grain. Wheat grains essentially contain a lot of starch. Bacteria will start to feed on the starch, and then microorganisms will start to feed on the bacteria. So what you're doing is you're creating a small food chain this way. This way, yeah, you're able to then see way more cells in the sample after approximately 4 or 5 days. See how this worm is kind of trying to feed off the biofilm growing on the plant. So you can often observe them do that. They have a little mouth, and then they will kind of scratch off bacteria, algae, whatever is growing on the surface. How many, it's a question, interesting one. How many species did go extinct in these samples, do you think? Extinct, okay, we have to define a little bit the word extinct. Extinct is a biological term, which means basically that the species dies off and is then basically gone away from the world. Yeah, I think what you mean is that how many of them actually have died off, and this does not mean that they have gone extinct. And I would say quite a lot of them, because this is a normal procedure. If you have a water sample standing in a jar, what you will be able to observe a certain progression of organisms. There are certain organisms that will start to increase in number, others will decrease, and some of them might actually die off in the jar, okay, or at least go down very much in the number. That is a completely normal thing that you are able to observe. Yeah? And this depends on a variety of factors, on the availability of food, the oxygen concentration is important, and so on. Yeah? And especially if your sample goes anaerobic, as I mentioned, basically if there's too little oxygen, then this is going to cause an explosion in certain bacteria. And all of the larger, more interesting organisms, they will die off. So those worms are going to die off because they need a lot of oxygen. Yeah. So always make sure that if you want to prevent organisms dying off, then always make sure that there is always a supply of oxygen present. Yeah. And bacteria are able to reproduce rapidly, even Some of them even in the absence of oxygen. Yeah? So the next question, why can't you just add some food to the entire sample? Why make an enrichment culture and add food? Okay, so the question is the following, why not simply take this tube and basically add a little bit of food? Of course I can do that, and however, you see, you never really know exactly what's going to happen and which organisms are going to start to reproduce. So it could be that some of them will start to really increase in numbers and otherwise others will decrease. And if you want to keep the original sample, then you should kind of take out a smaller amount here and then try to add some food to that if you want to preserve the original. Yeah? So this is generally something that I usually do. I will take a smaller amount out and then I'll try to enrich it so that in case something goes wrong, then I still have some of the other original sample left over. Now bumped into the camera. Okay, yeah. So this is kind of the, I hope I didn't forget any questions. Yeah, so this sample is also doing fine. I always have to check a little bit on the time. Yeah, so I would like to move on a little bit. And another, so this one I'm also going to keep. I'm going to keep the one from the marine water sample. So, and now this one over here, if you've been following some of my other videos, this might actually look familiar to you because this one really has changed quite a bit. Some weeks ago I found an old piece of wood with some lichens on it, and I added a little bit of water. Here again, I just want to show you, if you find something like this as well, you do the following: you add a little bit of water on the bottom. The water will soak up into the wood, and some weeks ago I found tardigrades on here, nematode worms, all sorts of interesting things. Because you do not want to have the whole sample submerged in water, because again this might reduce the oxygen concentration a little bit too much. But there's something else that has happened here, and that is it has gone moldy as well. So I don't really want to touch this here, especially with my little cut in the finger. So what I'm going to do is I'm going to carefully take it out with some tweezers, and I'm going to also try to see what is growing on here. It's actually quite a nice example of because some time ago there was not the fuzzy layer of fungus growing on it, so you see that there is a progression of microorganisms happening here. So I'm going to do that. How do I do that? I need some larger tweezers so that I'm able to hold this better, because this is a little bit disgusting. I will, of course, as always, collect some water and put it directly. You don't see that, so let's move this up here. Up, I touched it, so here. And let's try to now take some of the lichens here. And this is really a lot. Okay. And if I were to leave it on here, it would be way too thick again. So I'm going to simply take this off and put it back, if possible. Over here I have some fungus. I'm going to take some of the fungus, if I'm able to get some of it. Oh, yeah, it's kind of difficult. Yeah. I should also add a little bit of water there. All right, let's try it here. It's kind of difficult to collect the fungus, but maybe we're able to see a couple of spores. I don't know. I got some of the. So let's put this back, and 2 cover glasses again, and one. Let's make sure that this is the one here, and one here. Okay. So let's put it under the microscope again. And let's put it in here. So change of camera. So okay. And in focus. Yeah. And yeah, some time ago I found several tardigrades. This is a little bit a question of actually of luck. Yeah. But otherwise not so much of interest here. Whole bunch of debris. Probably many bacteria to be seen. There is a little bit of movement going on as well. So, but this is still at a fairly low power. Let's go up here. Yeah, mostly bacteria. Not so super interesting. Yep. Yeah. Mostly smaller microorganisms. Yeah. This here looks a little bit like an amoeba. Let's watch this a little bit. I just want to point this out here. The arrow. Where is it here? Yeah, this one over here could be an amoeba. You see this extension over here, so-called a pseudopodium, where essentially it starts to reach out. And sometimes you're able to see it's changing its shape as well. Yeah, of course. Yeah, here we go. Yeah, that's an amoeba. And usually those are also quite interesting to observe on the time lapse. Let's go up a little bit more with the magnification up here, and I need to focus. Yeah. Okay, well. Those amoeba are quite nice to observe, however, also sometimes difficult to see because they sometimes don't have a color. They just yeah, and their movement is also not always very fast, but you can now see that it's able to change its shape. Yeah? And yeah, maybe it's in the process of trying to engulf something over here. Maybe, yeah, it's trying to go around it, and maybe it's going to close on the other side, and then whatever is in here in the center is then caught. Yeah? It's a little bit like the white blood cells. Yeah, you can also see that those circular structures in here could be the nucleus. Excuse me, I've got to cough. Okay, yeah. And it's changing its shape again. Yeah, so okay, a little bit as expected. Let's go a little bit back again with the magnification. So, yep. Look, this one could be another one. Okay, I have to go up. Yes, this one actually is nicer. Actually, there are two of them. Actually, there are three of them. That's 4 of them. Amoeba paradise here. Okay. Need to maybe make it a little bit brighter. Yeah. So you have one here. You've got another one over here. One over here. Yeah. Maybe this one here as well. Yeah. But this one over here specifically, if you look at this one, you can actually see that it's it's starting to radiate out its so-called pseudopodia, and it's able to, this way it's looking for food. You know, this one down here as well. Yeah, see, it's reaching out. Lots of amoeba here. Yeah. How do we know if it's Naegleria? Okay. So that's the thing is amoeba are quite well known. Look how it's because it's in the media. Sometimes you can hear about the brain-eating amoeba, and do you know how many amoeba there are out there? I mean, it's a very large number. You cannot automatically, assume that it's of a specific species. You'd have to do more analysis. Could they be dangerous on here? Maybe, maybe not. You don't know, right? However, those brain-eating amoeba that you hear in the media or heard in the media, they occur in water samples at warm water, and this is simply not the environment. So it is highly unlikely. Yeah. Of course, you want to keep some always take care of general hygienics. Yeah. So this basically means, of course, I'm not going to try to avoid touching it, especially if I just cut myself, right? But there is no reason to automatically assume that it's automatically this specific brain-eating amoeba type. Yeah? because there's so many of them out there. And but can you be sure what it is? Of course not. That's why you have to, of course, like always, you have to be cautious. Yeah. Yeah. So I wouldn't eat this whole thing. Obviously, I mean, that's disgusting anyway, right? But yeah, and I wash my hands afterwards. Yeah. But you do not always have to assume automatically that it's dangerous, because you find pieces of wood lying around in the forest, you walk over it, barefoot, and it's not an issue, right? Yeah? So we are in contact with those microorganisms every day anyway. Is the amoeba that much different to a white blood cell, or is the white blood cell type of many amoeba maybe once living like us inside mitochondria? So the question is a little bit, that's an interesting biology question, is this amoeboid movement, the ability to change shape? Is this something, a sign that somehow the amoeba are related in a way to the white blood cells? Yeah, because they're also able to change their way. And as a matter of fact, the amoeboid movement was, in the course of evolutionary history, developed multiple times. Yeah, so just because two cells have a similar characteristic does not always necessarily mean that they are related. Yeah, so this is, in that sense, it's quite common in nature that certain characteristics developed independently of each other, right? Yeah, and so the amoeboid movement of white blood cells and of amoeba can be traced back to the movement of the sliding microfilaments in the cytoskeleton. So the mechanism might be similar, but this apparently developed multiple times. Yeah. That's a starch grain, a potato starch grain over here. Maybe this is still a contamination that was left over from because I rinsed the microscope slide. Yeah. So again, we've got one over here. Yeah. So let's have a look now at the other one, the one where I tried to scratch off a little bit of the fungus. I don't know if this is here, are we able to see some of the fungal hyphae filaments somewhere? I don't know. I'm able a little bit, but not very well. Let me change the filter setting again. Yep. That here is very typical of a fungus, okay? Those, long structures. Yeah, these are the long cells of the fungus. This one actually is pretty fine, I would say. Very thin. Normally, they're a little bit thicker. What I'm not able to see so much are spores. Occasionally, you also see spores. Yeah. Yep, but those, some other microbes, bacteria are also floating around. Yeah. But that is essentially a little bit as expected, not super exciting, but essentially as you would expect. Yeah? So there are basically the fungal cells are typical in the sense that they're kind of branched. Yeah? But I have actually seen fungi that are a little bit thicker, that have thicker cells. Yeah. So this one is not so interesting, but it kind of shows a little bit that on the same piece of wood, depending on which part you look at, you're able to see different specimens. Could this be? These could be some spores. Let me see. Yeah, that could be. Yeah, these things maybe. Yeah. Let me change the thing here. It's interesting that this is... I've got the wrong filter setting. Here we go. Yeah. Okay. Here now that's better. Look at these dots in here. Could be the nuclei. Okay. Many fungi essentially... I don't know about this one so much, you have to zoom in more. Many of the fungi are so-called aseptate fungi. This means there are no individual cells, but it's essentially the cells are kind of fused together and you have one long structure with hundreds of different nuclei. These are so-called the aseptate fungi. Yeah? So it's a little bit of a unique unique structure, because normally we always think that eukaryotic cell has one nucleus, but there are indeed cases where the individual cells are very long and have hundreds of nuclei. Yeah. So yeah, I'm going to now interrupt myself again and to have a look at some of the questions here. Are brain-eating amoeba a different species of amoeba, or did they just mutate or get infected? So basically, I think what you are trying to refer to, and try to make sense of this a little bit right now, is the question is it possible for an amoeba or any microorganism by doing some kind of a mutation, genetic change, to become essentially now a different species? Like for example, you have other amoeba, and then there is some kind of a genetic DNA change, and then all of a sudden it becomes a genetic, a more dangerous species. Yeah, this is not possible. Okay, this is called pleomorphism, and is a completely outdated view and theory, where there were some folks were thinking back in the day that maybe it's possible for one microorganism to change and to become another species. That is not going to happen, just like that. Okay, so changes can only happen over evolutionary time. So of course species can change over the course of evolution, but this takes a lot of time, but not within a short time where one species is able to flip over and become a different one. Okay, this is would be a violation of a biological law as well. So this is the thing. Could it be possible that because of some genetic change, basically certain microorganisms now become dangerous without becoming a different species? That is theoretically possible, of course. Yeah. And how would we identify a species? Well, for example, let's stay here. This is how in the world would I know which type of fungus this is here? And as a matter of fact, I know it sounds kind of disappointing if I say that, you won't. You might be able to narrow it down a little bit, but you're not able to, just by microscopic observation, you're not able to actually reach a species level. Okay, and why is that? Because microscopy alone does not really provide you enough information to in most cases to actually identify it all the way down to the species. Yeah? It's a little bit like asking, okay, I look at the color of a car, and then I would like to determine which type of car it is. That's not possible. Yeah? Yeah. Or by looking at a person, a human being, and then determining which nationality does the person have, or which languages does he or she speak. This one here, that's a pollen grain. Okay, that's very typical. That's easy. Yeah? In the water sample there was some pollen. It's a plant pollen. Very, very typical. Yeah? Okay, so there are a couple of other questions. If the Ialosoma reproduce by cloning themselves, is the genetic variation not degrading? And that is a very interesting and a very good question. So essentially, the whole question is there are many species in the world which are able to reproduce asexually by cloning, right? And the question now is what happens to now the genetic variability, the genetic diversity? And indeed, it is like this that. If this were the only way to reproduce asexually, then this would be a problem, because then indeed there would be too little genetic variability. But those animals and microorganisms, they all also additionally have a mechanism in actually increasing their DNA variability, either by directly exchanging DNA. Some microorganisms do that, or they actually do also have a form of sexual reproduction. So also Ialosoma as a worm will reproduce sexually as well to increase the genetic variability, but they will also do asexual reproduction, right? So you actually do need both. Yeah, and there are some micro animals like the rotifers which primarily reproduce asexually, and scientists were kind of concerned. Well, if they only reproduce asexually, they know that because they're all females, then what happens to the DNA, to the genetic variability? Yeah, and then they discovered, of course, that they have other mechanisms of direct DNA exchange. Yeah, so essentially, what I'm trying to say is in many cases there is a mix of both. Yeah, so my link has been removed from YouTube. If you're interested in the study, search for the title "Erythrocyte and Blood Antibacterial Defense." Thank you for the comment. I will have a look. Okay. Yeah, so the question is now, I understand, but how would we identify it? By getting its DNA? I would give yes. That would be probably the easiest way. You can, if you're a specialist, then you might be able to go down and you might be able to look at specific details of the anatomy, and then maybe you're able to indeed go down to, in some cases, to the species level with certain animals, but with microorganisms this is often not possible. So what you can do is you can try to culture the organism in a pure culture and do a range of biochemical tests. This is what I've done during my university study with bacteria. I had to culture them and I did a whole range of biochemical tests. However, I think the far easier way these days is to do a direct DNA analysis. Yeah, and then you basically copy paste the DNA sequence into the database, and then the database is going to spit out a percentage DNA similarity. And then basically what you hope for is you hope for that whatever DNA you got from your organism is not in the database. That's what you really hope for. You hope not because then you got something new, right? If it's already in the database, then yeah, somebody already was faster than you were, right? Yeah, so this is, I would say I'm just going to skip this one again. So this was basically the wood. And I'm just going to move on a little bit now. What else do I have? I've got some other samples over there. What time is it? Yeah, it's already 1 hour. Because I do later on, for just a couple of minutes, I do want to show you a little bit the slides that I made last week. Okay. The fungi usually unicellular or multicellular. Okay, that's another one. Essentially, what you do is many fungi are in the transition of being unicellular or multicellular. What do I mean with that? Is that one characteristic of multicellularity is that there is cell specialization happening. And this basically means that certain cells become more and more specialized. And depending on the fungus, you have more or less cell specialization happening. So I would say that it's a little bit of a question of definition if a fungus is single-celled or multicellular, right? Because, of course, we would probably think of a fungus, of a mushroom, as we see it, we would say it's multicellular. That is correct. But then again, if you are able to take individual cells out there, and then you're able to grow a whole new fungus. So maybe you can actually make a case that it also has, is single-celled as well. Yeah? So, and it depends on the fungus as well. Yeast, for example, is a fungus, and yeast are single-celled, right? So this is a little bit a thing here. So this is also a pretty old one, a pond water sample over the time. Essentially, algae started to grow. I see that there seems to be also some white. I don't know if you're able to see those white dots on the surface. There seems to be also some kind of mold growing on there. So what I'm going to try to do is I'm going to try to actually, take this a little bit and see what this is. Can't. So I got some a whole bunch of slimy unidentified material now on my slide here, which I'm going to put directly under the microscope, and then we're going to figure out what it is. Okay. It's also pretty thick. And again, I really don't know. Okay, so let's have a look. And the scope again, let's turn off the arrow and let's have a look. Ooh, huh? What? You know what this is? It looks like a decomposing insect. Oh gosh. Of course, there was maybe a tiny fly fell into it. And basically fungi started to grow and bacteria started to grow around it. Yeah. Okay. Yeah. Yeah, that's yeah, of course, there were of course lots of bacteria and fungi on the surface because this is a source of nutrients. Yeah, and yeah, yeah. So nothing, nothing completely unusual. Yeah. So, but let's pull out some algae here. Okay, that's basically what is that? That is it seems to be. Let's count the number of wings. If it has only one pair of wings, then it looks then it looks like a diptera. It's like a fly. It seems to only have one pair of wings. Yeah, down here. Yeah, so this is basically an insect that fell into the jar and died, and then it started to decompose. I was actually hoping for something, but a little bit unexpected as well. Okay, so that is but actually let's have a look at. Let's take this away again, and the desk view. So I just show it to you again. So if you have a look at over here, on the side here, this white dot on the surface of the water seems to be another one. So let's take a small sample directly here, and I dropped it. Can I reach this? So it is pretty large. Here it is. You always want to spread it apart. And cover glass goes on top. This is way too thick, of course. Yeah, but usually for a first quick check, it's enough. So yep, lots of filamentous algae. Difficult to focus because the sample is so thick. Yeah, but otherwise, let's go a little bit up with the magnification here. Yeah, this is not. Yep, algae, chloroplasts visible. Not so super exciting. Again, it's a fairly old sample. I might think actually, I've been thinking about dumping this one out and making a little mini terrarium with some moss, and things like this. Looks also nice because this sample is also not very nicely and decorative either. Okay, see if we're able to find anything here. Also not. You see, if you look at some of those cells, they're not even very green anymore. So this actually means that the cell's probably already dead. The chloroplasts have broken down. So the live ones usually they look much nicer in the sense that there are, they actually look fresh and green. Yeah. So for example, this one over here. These algae over here, these still seem to be alive. You can actually see that, they're quite nice and green. There are some movement going on as well. And let's see if I'm able to. Okay. Yep. See, as I'm focusing, I'm already touching the cover glass here. I'm focusing, and this kind of moves the water back and forth. Yeah. So these algae here are alive, and what you are also able to see is on the surface. Also very common, especially it also allows you to estimate the age a little bit. You see, there's a lot of stuff growing on the surface here as well. That's a biofilm. So this basically means that algae that are quite fresh, so to say, they do not have a lot of this biofilm on the surface. And algae that are a little bit, or any surface that's a little bit older, will have had enough time for bacteria and other microorganisms to grow on the surface. So this is a little bit also a sign that this specimen is not the most recent one. So so that is kind of the I'm going to move this away. It's a little bit of a lower magnification, a little bit. Yeah. So not a very interesting one. I might actually throw this sample out. Yeah. I'm going to maybe check occasionally also a little bit some of the sample on the bottom. Yeah, but you see this brown stuff here, lots of decomposition going on here as well. Yeah, not something totally exciting. Yeah. So let's move on, I would say. Okay, so this is a little bit what I expected. I would like to show you a couple of other things here. I just quickly always keep on checking the time. So let's this one we throw out, and I would like to show you. And now here we need a little bit of luck here, because this one is also almost half a year old already, but this one is thriving. Yeah, this one is also a very nice sample, and here I also but that's a little bit difficult to catch, because there are if you are able to see that, I need to do the following. I need to, let me see if I've got another one over here. We have a pipette. Yeah, I'm going to cut the tip of one of those pipettes here, and we're going to really try if we're able to take up some of the things in here. And I know this is now a lot of, that's a lot of water. That's a lot of water that we have here. And I'm not going to put a cover glass on top, but I'm going to put this directly under the microscope without cover glass. Very risky. And at low magnification. So let's see. I was kind of hoping to find, yeah. So that's an ostracod, and maybe I don't know if I'm able to find a few of them that are still alive. Yeah, ostracods are related to, are water crustaceans. Yeah, but the only thing that I see here are dead ones. I know there are a lot of them still floating around alive. Yeah, but they are not moving, because those that are alive are actually swimming around quite vigorously, but I don't have them here right now. Yep. So these are so-called ostracods. Yeah. But these all seem to be empty shells. Let me turn off the arrow again. Okay. Yep. Yeah. Okay. What I might do maybe next time is I might prepare a few of them, because I have to catch them out of the water sample. And they like to swim away quite rapidly. So yeah, nothing here otherwise, but I'm going to keep it in any case, because I know that there is lots of interesting stuff in there. So last but not least, so I'm going to move away. I want to show you now another one here, and this sample is also over 2 years old. And where is it? The desk view. This is vinegar, and these are so-called vinegar eels. Let's see how they are doing. They have been growing. I got the sample from a colleague, and he's growing them as food for his fish, aquarium fish. And I've also been basically growing them. Yeah, and added a little bit of sugar, some vinegar, and they're floating around, usually on the surface. And I'm going to hope that I'm able to find a few of those here. And let's see how they are doing. And because these are essentially, let me see. I hope that I got some. Yeah, here they are. Okay, so these are nematode worms, and yeah, always moving around, always happy and active, but probably also one of the more difficult specimens to observe because they're just moving too quickly all the time. Yeah. So why is it so blurry? Here we go. Yeah. So these guys are very yeah, very active, and basically I have been growing them in the jar now for, I don't know, almost 2 years. Yeah. So very easy to grow. Yeah. And yeah, some of them are larger than others, and yeah, what you can do, however, as well is I don't know if I'm able to see this here. It's difficult here. Let me check the other one. I've got 3 jars. Just a second. There is some. Let me try this one here. I'll also put it directly on the microscope. Then this is now from the very bottom of the jar. Yeah, here are a few more. How do you like this? Yeah, that's basically yeah, it's almost a little bit of like a nightmare. Let me put this away again here. Yeah, so yeah, I got them from the bottom of the jar, and yeah, here this is also limits their movement a little bit. And here we can actually see quite nicely also a little bit into the digestive system of those nematode worms. Yeah. So yeah, so they seem to be doing fine. Occasionally I did add a little bit of fresh vinegar and a little bit of sugar, and then they're going to be happy again for a couple of months. So to study those vinegar eels, one could video an I guess I have not videoed it yet, especially time-lapse. Okay, so basically slow motion video. That is, I think, the question, especially if they're moving too quickly. And I think the problem with that, one could do that, but one would need a pretty fast camera. Okay, and there is still a little bit of motion blur going on. But what you can try to do is I mean, I didn't try that. Maybe add a little bit of glycerin to those vinegar eels to slow them down. I tried that once, but this killed the vinegar eels because the glycerin osmotically dehydrates them. So you have to use very little glycerin, but you can try to do that. Yeah. Yeah. So that is yeah, quite an active, yeah, yeah, quite an active one. Yeah. Again, these are completely harmless. Those vinegar eels, and they are basically nematode worms that are grown as fish food. Yeah. And yeah, where do they occur naturally? So, for example, when you have fruits that fell from a tree, apples, pears, whatever, right? Then they will start to reproduce there, and they will essentially convert, the sugar also into acid, yeah, and break it down. Yeah? Where do they come from? Well, I suppose that they are spread maybe over eggs carried by flies, yeah. And then, yeah, that's actually quite a very active. Yeah. So let's move in a little bit higher with the magnification here. Okay, wrong. Here we go. Yeah. And yeah, here we can now have a closer look at the digestive system of those nematodes. I actually once read recently, a couple of months ago, that some scientists discovered some of those nematodes frozen in somewhere in soil, and thousands of years old nematodes, and they were able to bring them back to life again. Of course, I also tried that. I tried to freeze those, but freezing actually killed them off. Yeah, so depends on the nematode worms. So these, at least, they do not like to be frozen because this kills them. Yeah. Yeah. So, and again, a question. Can you tell me more about archaea? I have heard of them, but I don't know much. Archaea, well, yeah, basically are also known as so-called archaebacteria. These are prokaryotes, which essentially like to live in very hot and extreme environments. Yeah, archaea are essentially a pretty interesting group of organisms. They are prokaryotes, so essentially they're just like bacteria, but they officially are not called bacteria anymore. They're called archaea. Yeah. So also with the decomposing insect, you mentioned something about how many pairs of wings it has. Yeah. So basically, by if you look at insects, there are different types of insects, of course, and some insects have only one pair of wings. So, for example, flies and mosquitoes belong to that group. And then other insects have two pairs of wings. For example, bees, wasps, beetles, and so on. They have two pairs of wings. Yeah, and I simply had a quick look at that, but of course it was simply just evident right away. It must have been a fly. Yeah, just by the shape of the body. Yeah? But that's usually one of the things that you can look at insects is count the pairs of wings. Yeah? And I thought vinegar was an acid, a weak acid, I should say, but I shouldn't microorganisms not like acid. That's an interesting one. So essentially, acid. Vinegar is an acid. However, this in order to grow those vinegar eels, you use 50% vinegar and 50% water. So pure vinegar would probably be too acidic for it. And yes, well, it depends really on the organism. And generally, it is correct that essentially many organisms do not like acid. But just think of the bacteria Helicobacter pylori, which is a bacterium which is able to survive even the acid of the stomach, of the human stomach. And it causes ulcers, stomach ulcers. And when it was discovered, no bacteria is able to survive the acid of the human stomach. Well, Helicobacter is able to survive it, right? So essentially, nature is very diverse. And if you look hard enough, you're going to find a lot of interesting things there. They found bacteria in nuclear reactors, where the radiation is extremely high. How are they able to survive that, right? But yes, they found bacteria even there. So nature is very diverse, and I'm always amazed at where you're able to find life. Silly question maybe. Each time you remagnify, you use your diaphragm only or sometimes light intensity. Okay, so basically what I've got is, so this is a little bit a technical question. I just want to show this to you here. I've got down here my filter wheel. Yeah, and what I have to do because I've got DIC, I always have to adjust the correct. It's not a filter, it's actually a prism, right, that is included in there. And for the best result, I have to match the prism that I use with the objective, on the top. So that is essentially what I'm doing, right? And that's why sometimes it will go like this, even though I do not change magnification, right? But I have to essentially, when I change the magnification around, I have to readjust this down here as well for the best results. And then sometimes I will also, is down here, I've got the light intensity regulator. I'm also able to change the light intensity. So that's kind of a little bit of a technique. Oops, and now the camera, for whatever reason, died. That's interesting. Why did this happen? Did the camera that died off? You know what I'm going to try? Is I'm going to try to deactivate it and activate it again. Maybe I'm able to. It's back. Okay, here we go. I don't know why this happened. Okay, so what else do I have? Yeah, because it's already 1 hour 19, but I do want to show you some slides from last week as I promised, because there is some interesting stuff here. So I'm going to do away those worms now, and we're going to have a look just at a few slides, simply to complete last week's live stream a little bit. So I'm going to put this away, and I'm just going to show you now. First of all, this one over here, the insect which I mounted in UPROL. Yeah, so because there is something I want to show you here. Okay, so I simply drop the insect into the mounting medium, and what the UPROL mounting medium does is, it as I told you already, it clears the insect. It means it brightens it a little bit. And when we zoom in here, okay, again, then I'm able to show you a few interesting things here, especially here at the legs. So I need to open up the condenser to make it a little bit brighter. And what we're able to see here is we're able to look directly through the exoskeleton. And I'm going to point to a few things here right now. What you see here is these here are the individual muscle fibers, of the insect. So we're able to look through the outside of the insect into the insect, and because the mounting medium brightened it up. Yeah? And if you look very carefully, and I don't know if you're able to see this because in YouTube the YouTube resolution might not be good enough, but these are the individual muscles. But maybe you're able to see that there are also light and dark bands going across like this. Yeah? Yeah? It's almost a dark, light, so it's like a striped pattern. Yeah? And that is basically, that's striated muscle. So that basically is basically very typical of muscles, that they have those striations, and I'm kind of happy that I'm able to see those as well. And if you want to know a little bit more about those striations, I did make videos about where I put some moose meat under the microscope, or chicken meat under the microscope, and if you do that, then you're also able to directly see those nice striations. Yeah? So that is essentially very typical of muscle. I just wanted to show this to you here to illustrate that just by using proper mounting medium, we're able to actually look into the insect, because it basically brightens up the specimen. Yeah? Look at these legs here. Yeah? They've basically almost become transparent. Yeah? So that's the reason why UPROL mounting medium is the mounting medium of choice by people who do insect microscopy. Yeah? Also this one over here, and just last week, we would not have been able to look through it, but now it starts to become brighter and brighter. Yeah. So I kind of like this. You can also bleach the insect. You can use some lactic acid to bleach it, or a potassium hydroxide. So there are a variety of ways of how you can prepare them and actually how to brighten it up. Yeah. So, but that was only one thing. So this is basically, and I would like to now show you two other things. And I would like to show you, this is now the, if you remember, I put some the peanut skin, the brown paper like coating of a peanut. Okay, this is now mounted in PVA glue. Okay, and now have a look on how it looks like when I mounted this in UPROL mounting medium. The same thing in UPROL mounting medium. Look how bright it is. I actually have to go down with the light intensity. Okay, and we are able to see the individual cells. It's the same specimen, only different mounting medium. So a resin-based hydrophobic mounting medium versus a water-based mounting medium. Look how dark it is. Yeah, that's the glue, and that is, is it UPROL or UKIT? I forgot. It's UPROL. It's the same one that I used for the insect. Yeah. So again, last week, if you watched last week's live stream, it all looked very dark. Yeah? But over the course of this week, the mounting medium was able to actually reach all of the parts of the specimen, and that really brightened it up quite a bit. Yeah. So this is something I also wanted to show you, and the effect is even larger when we look at pollen. Just want to show this to you. So that is the peanut. And now I also prepared some pollen. Again, if you want to see the preparation, I recommend that you have a look at last week's live stream. So that is again the water-based glue. Okay. This is how the pollen looks. It looks generally okay, one could say. Yeah. Okay, and that is again Uprol. Just a second. Yeah. So that is the pollen. And now watch this. Different mounting medium. Look how bright it became. Same pollen. Yeah. So last week it looked as dark as the other one, but it simply takes a little bit of time to clear and to brighten up. Yeah. And I also have UKIT. The pollen I made also it is another resin-based mounting medium. So let me remove this here again. And UKIT, which is another very popular one. Yeah, also looks bright like this. Yeah. So I just wanted to kind of tell you that different mounting media are good for different things. And for example, like water organisms, algae, and so on, you probably do not want to mount in those hydrophobic mounting media because it's not compatible. You probably want to use the PVA glue, right? Yeah, but because of a different refractive index, it's the PVA glue again. Yeah, it looks quite different. Yeah, but then again, looks darker. But then over here, somehow some of them seem to have lost their shape a little bit as well. Yeah. So this is a little bit something I just wanted to mention again to kind of round off last week's topic. Yeah. So what I'm going to do now is because, oh my gosh, it's been already, 1.5 hours almost. I'm quickly going through hoping that I answered most of the questions. Yeah, exactly. Don't forget about the permanent slides. Yes, here they are. I remember I saw a lot of small circle structures in the nematodes. Very small, but a lot. Yeah, I don't know what these, I don't know exactly what these circular structures are. Okay. I know I'm asking a lot of questions today, but the last one is how exactly does Uprol clear the insect? That's an interesting one, and I think that the reason is because it has to do, that's at least one of the aspects, it has to do with the fact that it is able to infiltrate, so to say, and reach all of the parts of the specimen so that there is no air in there. Because if there is any air in there, then there is a large difference in refractive index between the insect and the mounting medium, and this causes structures to appear darker. I think this could be one of the reasons. Yeah, but whether there is actual bleaching going on, I don't know about that. Yeah. How many different prisms or sliders in your setup? Well, there is, I don't know, honestly. There's, one in here. Then there is also one up here. Oh, I don't know if you're able to see this. Where's this? Here. So look what happens when I turn this. You see? There is this, I'm able to change the colors around. Yeah. And if I and there is also a polarization system down here, which I'm able to remove. And then I've got a regular bright field. Yeah. And then I'm also able to adjust something over here. So there are several things to kind of, but usually what I will do is I will try to keep the settings mostly the same. Usually I adjust it to have this nice blue greenish background because it kind of looks nice. Yeah, imitates a little bit of water. Yeah, but essentially you can do whatever you want here, or you just basically remove this and then you have got regular bright field. Yeah. Which sometimes is actually better as well because you have the natural colors. Depends really what you want to do. I usually leave the colors because it's colorful. That's it. Yeah. Thank you. You have one set of prisms and a slider. I thought that you use a different. Okay, yeah. So what I'm gonna do now is I'm just going to call it quits again for the day. It's been again 1.5 hours. I wish all of you a happy new year. Hope that you enjoyed this session again. If you have any recommendations, please. That's the most difficult thing is always to come up with some topics to talk about. Okay, yeah. Sometimes I improvise as well, as you've seen today, a little bit with the blood that I put under the microscope. So I'm willing to, also take up any suggestions that you might have. So I'm going to wish you now all the best. Hopefully see you around next time again. And happy microbe hunting, of course. Bye bye.